Good morning. I'm Ellen Thomas. I'm a clinical geneticist and I'm the clinical director at Genomics England. And I'm delighted to be joined this morning by Professor Dame Sue Hill, who's the Chief Scientific Officer at NHS England, and by Chris Wigley, who's the Chief Executive Officer at Genomics England. Please do ask us questions via the Q&A function. We're looking forward to discussing as many of those as we have time for towards the end of this session. So this morning, we're going to be discussing the contribution of genomics to patient care in the NHS. And genomics is the study of our DNA. And we've discovered over the last couple of decades that in terms of the pattern of genomic variation that we all have, we all differ from each other a lot more than we'd previously anticipated. And a lot of this genomic variation is what makes us all different from each other, but it doesn't necessarily have a direct impact on our health. But there is some genomic variation where a particular variant or a particular combination of variants directly cause disease. And the goal of the Genomic Medicine Service is to use that information in the care of patients. So I'm going to ask Sue, first of all, to introduce herself and to talk to us a bit about the journey that the NHS has been on in increasing the use of genomics in patient care.
Thank you very much, Ellen, and good morning as well to Chris. And good morning to all of you listening to this showcase on genomic innovation. I'm the Chief Scientific Officer for England, as you've already heard, and my role in NHS England and Improvement is as the Senior Responsible Officer for Genomics in the NHS. And I just want to start by saying the NHS has had genetic services since its inception in the late 1940s, and our first laboratories came into being in the early 1960s, and they were at that time looking at whole genomes, but through looking at chromosomal patterns. They're important because the chromosomes actually contain our genes, and they are part of that essential instruction manual, and when things go wrong, they end up in causing disordered function and then disease. From an NHS perspective, we of course want to make sure that we have the best possible outcomes for our patients. We know that some people live with a rare and inherited disease, and sometimes it takes a long time for those individuals to present themselves to clinical services and to indeed to get a diagnosis. In terms of everything that we're pulling together at the moment, in partnership with Genomics England, we are trying to make sure that that diagnostic odyssey is minimised, but also that we expand what we look for in terms of the potential for genomics to influence the disease course for rare disease patients, or indeed to provide a diagnosis that may help families and other members of that family. But importantly, and something that's often overlooked, is that cancer is indeed the cause of a genomic mutation. And so part of our service is also to focus on cancer and to ensure that we undertake comprehensive genomic profiling of cancers to make sure that we can personalise and stratify medicines for those individuals. And finally, there are of course more common diseases in the population where genomics plays a part and where we need to find family members that might be influenced by that particular genomic mutation, or indeed adverse reactions to drugs that can cause quite severe reactions in some. So Ellen, to answer your question, and that was quite a long-winded answer for you, I think genomics is central to what we're doing in the National Health Service and is central to us improving outcomes for patients and their families.
Thank you very much for that comprehensive introduction. And I wonder, Chris, whether you could talk us through the contribution that Genomics England has made to some of the journey that Sue was talking about through the 100,000 Genomes project and now into the Genomic Medicine Service.
Absolutely. And as Sue was saying, the history of genomics in the NHS is rich and storied. I guess around the turn of the millennium, there was this big moment with the Human Genome Project. The first time that we had a full map of all of the 3.2 billion base pairs that are out there in the genome. And that, I think, opened up a vision of, if we can understand everything in the genome, how can we use that both for healthcare to treat patients and also for research to understand our bodies, what kind of drugs work, and so on. And Genomics England was created as a vehicle to really build a data set and a set of operational practices that would allow us to take that vision into reality in partnership with the NHS. And so, as many people are aware, the then Prime Minister in 2012-13, David Cameron and his family, they had a kid, Ivan, who sadly had Ohtahara syndrome, which is a genetically based or driven disease. And like many families, they became deeply expert in this condition and the science around it. And in conversation with people like Sally Davies, who was then the Chief Medical Officer, and John Bell, who was John Bell, the Regius Professor of Medicine at Oxford, came up with this idea around if we could sequence 100,000 genomes, and doing just one as part of the Human Genome Project had been a couple of $2 billion and quite a long time. So this had become much more affordable and kind of rapid at that point. We can really start to understand those variants that you talked about, which ones are common, which ones are rare, which ones actually drive disease, and so on. And so in partnership with the NHS, Genomics England went through this journey of the 100,000 Genomes Project, thanks to all of the people who participated and offered their consent for their data and tissue to be used for the project, sequenced 100,000 genomes, and through the NHS returned those results to the participants. And that really formed the basis of the whole genome sequencing part of the NHS Genomic Medicine Service today, which, as Sue said, is very wide-ranging and goes from single gene tests into exomes, which are all of the genes, deep read panels for cancer, and then all the way to whole genome sequencing today. And that interface of the healthcare delivery part of that service, and then de-identifying that data, making it available to research, and then using the insights from research to feed back into healthcare through the National Test Directory, which Sue and team own and update every year, means that we can really have a learning system that we can keep sort of learning, updating, and kind of moving those horizons forward for the benefit of patients and research participants.
Excellent. Sue, I'm aware that you've also been very involved in the mass testing within the NHS for the COVID-19 virus. And that's a context where we're also using DNA-based testing, but instead of testing the DNA of the people suffering from the COVID virus, we're looking at the DNA of the virus itself. And has there been an impact of that mass testing programme on the rest of the Genomic Medicine Service?
So from an NHS perspective, we've been both testing using a molecular test called PCR testing to actually identify whether the virus has been present. As part of the broader response to COVID, we've of course been using those sort of tests, that type of tests, for mass community-based testing. But one part that I've been really delighted to be part of is to establish the sequencing capacity that's been required to sequence the virus and to understand variants that may be circulating in the population, or indeed brought in from those people who've travelled abroad to countries where other variants may be circulating. And those may come back in and then start to be circulated within our population. It's required quite, us to think collaboratively, how we can work with the private sector, how we can stand up new facilities, but also how much we actually need to understand at a surveillance level the type of sequencing we need to do in terms of the numbers, but also how we support national studies. And those national studies are both reference studies within the population to help understand the relationship to vaccination or, for example, antibody studies, but also, and I'm sure something that Chris will come on to, is how it can be linked with eventually the whole genome sequencing of individuals that have been affected by COVID, that's been part of the genomic study. And we can come back on that hopefully a little bit later on. But to specifically answer your question, Ellen, which was about the impact of standing up that sequencing, and because of this rich history we've had in the NHS of using genomics, of bringing in new technology, of using whole genome sequencing now in a partnership with Genomics England. And part of the reason that partnership was established was because of the size of the data sets, the ability actually to analyse those 3.2 billion letters in our genetic code, and to do that in a way that's rapid and can be fed back to patients. So some of that expertise has been in the NHS has been used to help establish the sequencing capacity that we've needed, because we all know the number of positive cases we've actually had in the population.
And I wonder whether the greater awareness possibly across the NHS and beyond of the use of molecular tests in healthcare has really been driven in part by that mass testing programme as well.
I think there's no doubt about it. The British public now know about molecular testing, they know about PCR testing, they know about lateral flow testing, but they also know about the importance of sequencing to find variants. So although that might not necessarily translate across into human disease, I think with the outcome of studies like Genomic, and with our ongoing, I think, joint approach to making sure that we upskill the British public in terms of their knowledge of genomics and how it can contribute to health and also better health and outcomes, it's all coming together in a way to create a perfect storm.
Absolutely, and I'd really like to move on now to another context where we're using sequencing as an expansion area within the NHS, and that's thinking about cancer treatment and cancer care, where we are sequencing in this context. We're sequencing the DNA and the genome of the tumour itself, and it's the changes in that tumour and the changes in the genomic pattern within that tumour which may have driven or caused the tumour to spread. So in the context of cancer care, Sue, I wonder whether you could give us a sense of how genomics is having an impact on patient pathway.
So of course, from an NHS perspective, we have to think about how we can support the some 350,000 people that get a new cancer diagnosis every year, as well as those that get a reoccurrence of that cancer, and to think about how we best bring in genomics more comprehensively across that population. Part of what we've been doing, though, is moving towards, and Chris mentioned this earlier, which is to establish a much greater gene panel, so number of genes that we look for, or molecular targets, as we often refer to them in cancer in particular, that might be both DNA based, so part of that instruction sort of manual, but also part of the translation, which is the RNA side of that functional genomics pathway. How we do this in an affordable and more rapid way. So we've been introducing 500 gene panels that are being applied in what we call a pan-cancer approach. So they contain all the targets for all the tumours that we see within the NHS, and also all the targets that enable treatment to be stratified. So by that I mean that different treatments can be used for different people in terms of the molecular signature that they display. But what's really important is that focus is on genes. What we've known from the 100,000 Genomes project is we have somewhere around 25,000, and there's always a number where it could be between 25,000 and 30,000 genes, but also the genome, with all those letters that Chris mentioned, that there are parts of the genome in areas that we call non-coding parts of the genome that are almost like areas that we haven't really read properly before. And so part of our strategy is to introduce more whole genome sequencing in cancer, because we think in addition to those 500 gene targets on panels, we think there's additional information that we can gauge from sequencing, whether that's in the sequencing we do today or whether it's in a different type of technology that enables us to look for very specific changes in that cancer tissue genome.
Yeah, and it's incredibly exciting now to be at a stage where we do have this kind of range of tools at our disposal, as Sue says, to kind of pick from depending on the situation. And just to pick up on the point about what we can learn from whole genomes in the sort of non-coding region, we're starting to understand that what back in the sort of 1970s and 80s was sometimes referred to as junk DNA, so this DNA which is not in genes and coding for proteins, which is 98% or so of the whole genome, that actually signals in that DNA, whether it's variations there or rearrangements and structural changes, can actually tell us a lot about whether an individual person is going to respond to a given treatment or what the best approach might be for them. And I think, like all of our work, it really comes down to the sort of human stories that we can influence and hopefully improve through this work. So it's incredibly exciting that today, through this partnership of the whole genome sequencing program in the NHS Genomic Medicine Service, any kid or young person who gets cancer in the country will be eligible for whole genome sequencing. People with blood cancer, which is often sort of very aggressive and challenging, will be eligible for it, and other types of cancer as well. And with Sue and her team and the test directory kind of expanding the indications that are eligible for this, we're hoping that we can bring sort of diagnosis and personalized treatment to more and more people. And I don't know if we want to go into it now, but I think it's also a great example of where research and clinical activities are really kind of coming together.
So what I was going to, what you just prompted me to say and to tell the story of was an 11-year-old child being seen and treated at Great Ormond Street. And through whole genome sequencing it was a hematological cancer, a blood cancer. And that although other tests had been done and usual treatment given, the patient wasn't doing very well. And whole genome sequencing revealed a particular type of blood cancer, myeloproliferative blood cancer, that enabled that individual to access treatment they wouldn't have been able to access before, and they're doing very well. And we've had really good examples of children with brain cancer. Through the 100,000 Genomes project, 50% of those 18,000 patients with cancer who participated had an actionable outcome. That was either about leading to a change in their treatment or accessing clinical trials. And I think that's a really important part of where we're going in cancer. But of course, from a National Health Service, we have to be in a position to offer the most appropriate test for that particular condition, and that's why we have experts who work with us to determine what tests should be used for what type of, whether it's a rare disease condition, a common disease, or whether it's a cancer.
Yeah.
I'm very interested in what you've both said about the technologies that are being used in the cancer testing with a combination of the panel tests and the genome sequence tests, and the approach that the NHS is taking of really streamlining and mass producing the sort of wet lab phase of those tests in order to get the maximum value and the maximum robustness from those tests. But then that process of the expert clinical scientists within the NHS really delving into that data so that the data production becomes pretty routine, but the expertise required to delve into that data and to produce the personalisation and the individual insights is then the key value that the NHS is adding to that data, which is really contributing to patient care. And that's a very interesting model.
Yeah, and just to pick up on that, and this point that Sue and I have both touched on about, I guess, this innovation pipeline in cancer, it's a really good example of how the worlds of healthcare and research are so linked here, in that people who are coming through the whole genome sequence service within the Genomic Medicine Service are asked if they would like to participate in research. And obviously, particularly for cancer patients, that can be incredibly relevant for things like clinical trials, like Sue mentioned. And we're finding that a very large proportion of them, the vast majority of people, are opting into that, which is fantastic. That then means that we can work with those participants to answer questions which are just at the sort of edge of what we understand in this field. And Sue and I co-chair a group called the NHS GMS Research Collaborative, which is quite a mouthful. But what that does is it brings together the work that's happening in the NHS with the Genomic Medicine Service, with the National Institute of Health Research, and with Genomics England, so that if researchers in academia or in biotech and pharma are doing work in this space, there's a single front door, so to speak, to come to be able to access those assets in an appropriate way, do the work on the research, and then bring those benefits into the clinic. And one proposal that we've had just recently, which is, I think, a really good example of this kind of exciting ability to innovate just at the edge of what we know, is exploring the question of, can we use whole genome sequencing insights to understand and kind of triage who, for a given cancer, will benefit from chemotherapy and radiotherapy, or who won't. And we know that from a patient perspective, many of us will know someone who's been through chemo, really horrible, long process, lots of pain and suffering involved. But if it's going to kill off the cancer, absolutely worth going through that journey. But if you're a patient who's going through that journey and it doesn't impact the cancer, that's just really painful and kind of unnecessary. And also, to the point about affordability, it's very expensive for the NHS to give these treatments if they're not actually going to benefit anyone. So, question mark, can we establish through looking at someone's genome, okay, you're one of the people who will actually benefit from this, and so it's going to be long, it's going to be painful, but it's going to be worth it. Or actually, you're not someone who's going to benefit from this, so we'll take a different approach. We'll maybe explore what kind of clinical trial we can get you onto. And that kind of, where we can see those insights from research coming straight into benefiting patients who are going through the service. I think it's just incredibly exciting time and kind of space for these technologies to be really coming to bear in the clinic.
I just wanted to bring up, I guess, a couple of points in relation to that, Chris. And I've made actually no secret that I've had breast cancer. I was a participant in the 100,000 Genomes project. I learnt that actually my breast cancer was genomically quite quiet. But as we move forward, what we have the potential to do, if we as we will be doing, saving those whole genome sequences and their analysis, we have the opportunity to go back and look at it again when further evidence becomes available. And that's the same for rare diseases and any of our common diseases. So that is really a positive benefit from whole genome sequencing in terms of it is something that keeps giving. But the other point I wanted to make was from the 100,000 Genomes project in particular, this alignment between routine clinical care in the NHS and research has so many benefits. In the 100,000 Genomes project, we returned 141 diagnoses to patients that wouldn't have had them if there hadn't been academics and researchers from industry looking at the more complicated variants that didn't necessarily have all the evidence to bring them into the clinic and be fed back to the patient at that time. There's another 131 still waiting for that evidence to be generated. But it just shows us what we can do as we go forward. When we have what we call variants of unknown significance, where we have to establish maybe other experiments to determine whether they are indeed pathogenic, they would cause disease in that patient, or to look at all the data that's been collected across a person's life course to understand whether it would fit with all the characteristics that they're presenting. And the other thing, Chris, and I think people often don't realise this, is how we've used the learning from what was the 100,000 Genomes project at that moment in time, but will be through this alignment between ourselves, through the NHS and Genomics England, how it's enabled us, for example, to design our rare disease gene panels.
Yeah.
What do we need to introduce in the NHS to systematically investigate, let's say, hematological rare diseases or those associated with cardiology or with neurological presentations. It's enabled us to understand how we need to standardise an approach to reporting and interpretation. And part of this, in the NHS, our principle is equity of access, driving the same outcomes for everyone, wherever they live, whether they live in Whitehaven, Penzance or Goole, for example. And I always pick quite extreme examples because I think that's what the National Health Service is about, and that's what genomics has to be about as we work together, driving that levelling up agenda, but using everything we can to ensure we have the best possible outcomes for our patients and our population.
Yeah. And it's a great example, Sue, of how that kind of infrastructure and machinery means that this sort of learning system that we've talked about really kind of comes to life. And I think it's a place where Great Britain is really well positioned to lead because of the nature of our National Health Service. And it means that when a smart researcher finds something out in, could be Oxford or Cambridge, could be Manchester, could be Newcastle, could be Penzance, right? Could be, or Exeter.
Or Goole.
Or Goole. That research can feed into the system and then be available to everyone across the whole country.
But we have to do it in a controlled way, because what we learnt that previously, and one of the reasons we built upon the learnings from the 100,000 Genomes project and other evidence we had in the NHS, was that we have to be systematic. But we are unique. We're an integrated healthcare system, although some people would say it's semi-integrated, which in actual fact it probably is if we start to look down at a primary care level. But we have, all of us, we have longitudinal health records that can actually shape the type of characteristics we might present at any given time that will contribute to the overall relationship between, you know, what we see in in terms of genomic testing or our DNA or our RNA, and indeed what is presented by us in terms of abnormal function or other types of symptomatology, often referred to as the phenotype, so that genotype-phenotype interaction.
Absolutely.
I think that leads us on really nicely to the world that I inhabit mostly in my clinical life, which is seeing patients who have rare and inherited disorders. And I think one of the big developments that we've seen through the 100,000 Genomes project and into the Genomic Medicine Service is the ability for us to really cast our net widely and do a broad genomic test. So we often do what we call a trio exome or a trio genome. So that's where you often sequence a child with a developmental disorder, and both of their parents and compare either all of their genes in an exome sequence or the whole of their genome in a genome sequence. And that really enables us, when we strongly suspect that there is a genomic disorder, it enables us, but if we can't precisely pin it down by examining the patient and by talking to the patient, we can really do a broad test. And then once we've got the data back, that's the point at which the clinicians and the clinical scientists can get together and throw their brain power at that enormous body of data to see whether we can really pull out the high value healthcare information out of those broad genomic tests. And to be able to do that in the diagnostic context and not just when there is a research project that happens to be available to us in a particular place at a particular time has made a huge difference to the way that we look after our patients. And I know that, Sue, there are a number of different services within the NHS, within the Genomic Medicine Service at the moment, with specialist laboratories offering these sorts of broad tests in a number of different clinical contexts. And I just wondered whether you might tell us a little bit about some of those clinical contexts and what sort of impact that care is having and the personalisation that can really result from those tests.
Yes, so we of course have whole genome sequencing in rare and inherited conditions that is driving that triome-based approach and trio-based approach. I'm not making up a new word here, just everyone, just to be clear. But I think it's worth remembering that both in the 100,000 Genomes project, over and above standard of care, so what had been, exhaustively tested for in the NHS, when patients came into the 100,000 Genomes project with rare and inherited disease, we saw what we call a diagnostic yield, so a diagnosis returned in about 25%, but that went up to 50 to 60% in some conditions, so quite condition specific, which is what we would expect actually. Yeah. It's not unusual. Yeah. Now with the current live whole genome sequence service in the NHS, which is a world first. Nobody else is doing it in the world. And that's why this partnership is so unique between our two organisations. We are already seeing in some diseases like cystic renal disease, so a particular type of renal disease, we're seeing diagnoses being returned in one of two patients. I mean, it's just absolutely phenomenal. And that is going down to a diagnosis being returned in 1 in 3, for example, for some other conditions. But what we have done from NHS England is establish 3 national services. One is a national exome service for seriously ill children and babies, so they're often referred to as NICU and PICU, and that's a national service run out of Exeter, and we've had around 1,200 referrals to date, and we've got a diagnosis in about 40% of those, and that's resulted sometimes in changes in treatment that have meant that those babies or children have left intensive care units. Sometimes previously they may have been there for 2 years until this has been established. Really astonishing. We have a national fetal exome service that is really truly world leading, run out of Great Ormond Street and Birmingham, and that's giving families back diagnoses, again at that similar sort of level of 30 to 40%, where they can make very difficult decisions. The same with the NIPT, the non-invasive prenatal testing service that we're trialling in 3 organisations at the moment, that helps in terms of identifying whether a pregnant woman is carrying a baby with a high likelihood of Down syndrome. And I have to say I'm really grateful that we've done that, because one of my nephews, his wife has just had that test. And so it's a real comfort. Yeah. Or in sometimes, part of a heartbreaking decision, that those sort of tests are now available more systematically.
And I think, Sue, that focus on the human stories is so powerful, right? And I guess almost to place some of that in context, we've talked about doing trios, right? The patient themselves, their mum and dad, to try and understand what's happening. And indeed, this was actually the case with the Camerons, with Ivan and Otara syndrome. The big question was, if we have other kids, will they also have this combination? Is that 100% chance that we know that if we have another kid, they'll also have this variant in their genome? Is it a 50-50 chance, or is it actually what we call a de novo mutation in that genome? So it's actually a 1 in a million or less chance. And those kind of human decisions about family and so on are so profound. And also, to Sue's point about diagnosis, just a couple of, I guess, contextual points there. One is that the baseline before these kinds of programs was that people would often be left without a diagnosis for years, for decades, or for their whole life, a disease without a name. And actually just getting to a diagnosis is so powerful. And I remember one of the families from the 100,000 Genomes project had a son, Owen, who had an undiagnosed developmental condition. And the family were kind enough to talk about their story on a video. And Owen's mum speaks incredibly eloquently about the power of getting a diagnosis. And it's, again, such a human thing. She says, I was so relieved because I thought it was something that I had done wrong. I thought it was something that we had done wrong. And actually, we know it's just this glitch in the genome. And as humans, we could just have this ability to kind of punish ourselves or to kind of tie ourselves up in knots. And actually having that diagnosis and saying, right, now we know kind of what the enemy is, so to speak. And that allows you to organize with other people who have the same condition, to create communities, to lobby for research into diagnoses. And of course, to, as Sue said, the benefit of having the whole genome on file and able to go back to through the sort of research library is what we sometimes think of as the kind of hopes and dreams factory part of what we do at Genomics England. That there are researchers out there working on these conditions who can then potentially a year, 2 years, 3 years down the line find out something about that case. And there are biotech and pharma companies working on developing treatments for those conditions. And that's hopefully how the magic happens, effectively, how we keep making progress against these terrible conditions.
I think though, Chris, part of this it's all come together, our ability to interrogate large data sets, to analyse them, the change in the technology. And so a part of this is how we ensure that the innovation is adopted. But also part of this is how do we spread this out across the whole of the NHS into all clinical specialties, rather than the specialists like Ellen who work as a clinical geneticist really looking at rare diseases. But what we know is 1 in 17 people in this country will develop a rare disease. I think that's an underestimate.
Yeah.
And so part of what we're doing in the NHS is to establish NHS Genomic Medicine Service alliances to really drive the embedding of genomics across the NHS, across all the different medical specialties, but using human stories, whether it's stories that you've told, whether it's about Jessica, who was another contributor to the 100,000 Genomes project, and her mutation that showed she just needed actually just a special diet, or the people we're diagnosing with monogenic diabetes who don't need to be on insulin anymore and can resume their lives, as HGV drivers or whatever they want to do, or how we're linking inherited retinal diseases with the ability to treat with gene therapy so they can see again. I mean, this is just, we're at a moment in time where everything's come together, but the pace will continue to accelerate as we're able to do more analytical techniques that you and the team will be leading, Chris, whether we're bringing in new technologies that have industries being developed, and where we use people like Ellen and others to help us really understand what's happening, and that we convert those messages to people in our healthcare system who actually are not specially trained in very highly complex genomic conditions. And I think this is going to be an extremely exciting next decade, actually, and beyond.
Absolutely. And it's interesting, all of the data, the technology, the analytics and machine learning and so on, as you say, are all coming together in these incredibly exciting ways. And I was just looking at the laptops which are around us in this room and thinking the database that we kind of safeguard at Genomics England on behalf of the country, the National Genomic Reference Library, now has approaching 60 petabytes of data in it. And if we think about a sort of high-end laptop that someone would buy for themselves these days, maybe half a terabyte, so a couple of 100,000 laptops worth of data, which probably would stretch the length of the country. I'm trying to think how long that is. So the data sizes are kind of galactic, and we need to use all of these complex bioinformatics and machine learning techniques to spot that slightly longer or slightly shorter bit of hay in the haystack, or the slightly kinked bit of hay in the haystack. But then we really need to do a lot of work on how do we communicate these incredibly complex kind of statistical and mathematical insights in a way which is relevant to a clinical team, like a doctor, a clinical geneticist, a genetic counselor, or whatever, and also to a patient and their family and so on, so that people can actually act on this. And I think that we need to keep working on how we interpret, share, communicate these incredibly complex insights, as well as the kind of the wizardry that to some extent can sit sort of behind the screen. Yeah.
And I totally agree with you, Chris, because behind all of this, there is an individual.
Yeah.
There is an individual with a family, and I know we are all really totally patient-centric in what we do, and that's what we need to continue to do. Yeah. It's how we can influence individual lives and those of their families and carers.
And sorry, you probably will think some questions. We're getting over enthusiastic, but we should touch on trust at some point as well. I think that's...
Yeah, no, I was just going to actually bring in, pick up on one of the examples that Sue used, which is the polycystic kidney disease example. This is actually very much at the common end of the spectrum of rare disease. Around 1 in 500 people in the UK have polycystic kidney disease, and this is a condition which causes patients to develop kidney failure and often need a transplant or need dialysis at some point around middle age. So it's a fairly significant cause of illness in the country, and I think it's a very interesting example of a lot of the things you've just been describing. The principal gene that causes polycystic kidney disease is extremely complex. It's technically very tricky to look at that particular gene, and that has really limited the number of people in the being cared for in the NHS who have a genetic diagnosis for their condition. We've assumed until recently that we needed to manage people without a genetic diagnosis in most cases. But that's really changed now, because we can examine the gene using whole genome sequencing. And so we're now really scaling up that testing. And that's been a two-way driver with the fact that there's a new treatment called tolvaptan for polycystic kidney disease, which can delay the progression of kidney failure in patients who have particularly aggressive forms of the condition. And the genetics contributes to understanding who will benefit most from tolvaptan, and the tolvaptan protects people's kidneys as they go through their middle years. So having those genetic diagnoses is contributing to that treatment, but it's also helping people to understand who in the family who in the next generation might develop kidney disease. We, at the moment, if we can only scan people, then we have to wait till the age of 30 to know whether people are definitely free of the condition, and do regular ultrasound scans. And when you're talking to families who have small children, maybe primary school age children, and you have to say to them, well, we won't know until these children are 30 whether they definitely have this condition or not. That feels like a long wait to parents. And if you can do a genetic test to work out which children need their blood pressure monitored and need their kidney function to be kept an eye on, and which don't, then that makes a big difference to families. And also for people who are considering extending their families, it then opens some of the options that Sue was talking about, including pre-implantation genomic diagnosis, where you can only reimplant embryos that don't have the condition, which is also available within the NHS. So I think it's just a really nice example of the ecosystem of the way that the technology, the pharmaceuticals, the patient experience, and also the implementation within the NHS have changed in recent years. And I'm just in the process of really handing over the management, the genetic management of the patients that I see with polycystic kidney disease back to the kidney doctors and the kidney clinics, who are picking up all of the basic elements of that management and taking on the outputs of these terribly complex genomic tests and really understanding what that means for their patients and how they explain that to their patients, and when they should phone a friend, when has this got complicated and we really need somebody who's more expert in the genomic aspects of this condition, and when is it actually we're fine managing this in the kidney clinic. So that just feels to me like a sort of vignette of really a lot of the elements that you've just been talking about.
And as is Alan, as you know, because we're working together on this, familial hypercholesteremia, which is an NHS long-term plan commitment, because this is a condition associated with a high circulating level of fats in the blood, associated with, an excess number of deaths, particularly in men below the age of 50, but that can be treated by drugs such as statins. And the community in general have not always accessed genomic testing, but also there's a big population out there who are sort of living with familial hypercholesteremia who've never actually presented into their health system. So we know that this we can test for it, we can treat for it, but it requires a particular way of working to ensure that we can find the case that has the high circulating fats in their blood, and then look at all their family members to understand whether they're carrying it, or indeed whether they can start early treatment, and that includes early treatment in childhood to prevent cardiac events. So that there are many areas that we can take that more systematic approach to.
And I think, Ellen, your sort of vignette also, I think, paints a great picture of what mainstreaming these kind of techniques looks like, right? You're saying, I personally don't have to do the details of each of these cases anymore. Actually, the mainstream clinical teams can deal with those. And I sometimes get asked about, okay, well, if we're talking about levelling up, we're talking about a national scale service. Do we have enough, clinical geneticists? Do we have enough genomic counsellors, etc.? The technology systems to some extent can scale to infinity, but there are actually a lot of really important humans in the mix as well. And Sue, this is very much your area, but I wonder whether it's worth touching a bit on if we're bringing genomics into the mainstream of health, how do we actually both bring the existing people with us, and how do we have enough of those people to actually do this at scale?
So, Chris, really good question, and I think we're having a great discussion here. We have, since the start of the 100,000 Genomes project, actually, we've had under Health Education England, we've had a very focused education programme to start to develop resources to essentially upskill the current workforce in the use of genomics. But of course it has to be more systematic than that. We have to embed it in both the undergraduate and postgraduate training, particularly of doctors. And so we're working very closely with the Academy of Medical Royal Colleges to ensure that it is in the curriculum, the postgraduate curriculum for all the relevant specialties. But again, also part of this is bringing it to life, because if an individual clinician in a particular specialty is only going to see a patient with a potential genetic cause for their disease once every 3 months, but they need to continue to look for it all the time, then we need to ensure that the education and training is right for that particular intervention. So we're doing a lot of work at the moment on individual pathway pieces of work, so that we understand what both the just-in-time education is required, what sort of knowledge we can make available electronically, so people can go and get their quick fact sheet, but also how we can think about the type of support we need to give to patients or family members, especially when they're getting a rare genetic disorder where they might be the only case in the country, or in a very small number of cases, where they really don't know what the impact would be on having other children. Or it is such a sort of severe diagnosis, essentially, with you know, for example, you know, a child dying before the age of 5, which is what happens in many rare disease diagnoses, then we need to work on ensuring that our broader healthcare team have those genomic counselling skills. And so part of this is transferring some of that skill base into our nursing workforce, and that's why we've got a big nursing and midwifery transformation programme in NHS England. But we will never stop needing to do workforce education and training interventions. We're always going to need clinical geneticists because they need to help with those really complex cases and support the rest of the clinical teams. And that includes our clinical teams in cancer, because they've not always been used to receiving very complex genomic information. That means they may even have a choice of the treatments that they do. And we've learned a little bit about this by rolling out something called DPYD testing, which is testing to know whether patients who receive a particular type of chemotherapy called the fluoropyrimidines, whether they are going to experience a severe reaction and sometimes fatal reaction. And indeed, through the rollout of this across the NHS Genomic Medicine Service, we've stopped several potential fatalities in early testing, early understanding of whether those agents need to be used or not.
Yeah.
So this is such a widespread field. But again, just coming back to what I've said earlier, something that is life-changing both for the individuals, but can also be quite changing for the clinical practice of our clinical teams.
Yeah. We had Eric Topol on the G-Word podcast, who was lobbying for every medical student should sequence their own genome and use that as a sort of study aid through their course. I'm not sure if that's part of the vision for medical education, but I think it's an interesting idea.
It's an interesting concept, one we shouldn't actually throw away at all.
I wonder whether we could just come back to something that Chris said earlier about talking about trust in this area. And Chris, one of the big responsibilities of Genomics England is holding and curating the National Genomic Research Library, which holds the genomic and healthcare data of the participants from the 100,000 Genomes project, from other research projects, and patients who are having their genome sequenced as part of the Genomic Medicine Service are offered the choice as to whether they would like to donate their genomic and healthcare data and join the research library. And you've talked a little bit about the size of the data and the challenge that poses, but I wonder what other challenges and also excitements really go with that data responsibility that Genomics England is holding.
Absolutely. Well, I think the first thing to say about the data that we hold is that it's not our data. It's the data of the patients and participants who've kindly volunteered to share that with us and through us with researchers in academia and in biotech and pharma, and that places a big responsibility on us. The reason that we've called it the National Genome Research Library is this metaphor of a research library. So at university, I did a slightly unusual combination of computer science and medieval history. But for the medieval history bit, I used to go to these obscure libraries where they have all of the old manuscripts, and you get given white kind of gloves, and you're not allowed to take pens in and so on, just a pencil, and you can't take the books anywhere. And that's very much the model that we have with the trusted research environment. You don't have to put on white gloves anymore, but you come in, you can study the data, you can run algorithms, you can do analytics, and you can take out your findings, you can take out the insights, but you can't take the data with you. The other analogy that we sometimes use is the aquarium. You can come in, you can admire the fish, but you can't take the fish with you. It's not a fishmonger's. And so that model is one of the ways in which we try and act in a way which is trustworthy. Why should people trust us with their data? Because we care about it. We're thoughtful about the systems. We're thoughtful about the technology, and so on. But the other big part of this is around empowering the people whose data it is to make decisions about who gets to access that data and on what basis. So we have a group called the Access Review Committee that when we get a proposal from an academic or a biotech or pharma researcher wanting to do some research, that group includes representatives from the participants whose data it is, and they have an absolute veto if they don't feel that the researchers have demonstrated that they have the right ethics in place, that they're answering a question which is going to be relevant and meaningful. They can absolutely say, you know what, go away, sharpen your pencil, think about this research project, and kind of come back when you've thought about it, which is, I think, a really powerful model in terms of not just engaging with patients and participants, but actually putting them in the driving seat around how we do this. And that means that over the course of the 100,000 Genomes project and the work that we're doing now with Sue and others, we've had more than 100 private companies access the data. We've had 4,000 academics register for research in there. And we haven't had, touch wood, and we're always working hard to sustain this, but we haven't had the kind of issues that we've seen in some other places of people being suspicious about motive or about the way that the research is being done, because we've empowered the people whose data it is to make those decisions. And I think that's absolutely vital, because to your point earlier about the ecosystem coming together, and we need to get both to diagnoses but also to treatments, we have to work with the private sector, as Sue was saying, to do that. Where do treatments come from? They come from biotech and pharma companies. But the how is as important as the what. And we have to do that in a way which is controlled and transparent, and where we're putting patients and participants, right in the driving seat of how we run those collaborations.
Of course, Chris, it starts with the patient, doesn't it? And their family right at the very beginning of their journey through a clinical service and into testing. And the work that we've done together, and particularly with Ellen here, we've done and spent a lot of time on ensuring that individuals can make an informed choice about both their data being shared to ensure that they get a diagnosis. If they are likely to be in a small group of people in the whole of the UK or in the whole of the world, to be able to get a diagnosis, it means that clinical data may need to be shared. And we've actually seen some really good examples where individual genomic data, especially in paediatric cancers, have needed to be shared with clinicians in Germany to understand whether they could access a particular type of inhibitor for that particular type of cancer. But it's both understanding that data will need to be shared for a clinical diagnosis, but also to give them enough information to make an informed choice about their data being used for research. And we've done a lot of work to make sure that we've got materials that are acceptable, both to individual patients or, in the case of very small children, to family members. And we'll continue to need to do that. But I think what we've done with the rollout of the live whole genome sequencing service into the NHS, and with all of our trio exome services that we've talked about earlier, we've made time available through people who understand how genomics are going to be used in their care and indeed research, to have those conversations with them, to answer their questions. And I think as a result of that, in our live service at the moment, Chris, those who've had that conversation, 93% have given their consent. I mean, which is a tremendous amount. And I'm always of the opinion that people who access our health system usually want to not only get their own diagnosis, but help others.
Yeah.
And I think that's really demonstrating that, but also the power of having good information, good trusted environments where the data is going to sit, but also the opportunity to provide them with information and support.
Yeah.
So I'd like us to move on now to address some of the questions that have come into us from our community today. So the first question, really, perhaps Sue, you could address this one first, is follows on very much from the conversation we were just having. Anna Dickinson and others have asked, what role will public trust and engagement play in ensuring that patients and their families actively participate in the service and research?
So I'm really delighted that question's been asked, because I realised I didn't say anything about it in my answers to date. But from an NHS England perspective, we've been very clear that at all levels of our system, so we have 7 genomic laboratory hubs, 7 genomic medicine service alliances, sort of then national sort of programme management type of committees. At all of those levels, we've insisted that there is patient and public engagement and involvement in all the governance groups, right up to the national governance group. And we have nationally a patient and communities forum that includes national organisations as well as representatives from Genomics England's participation panel. I think we can never do enough to involve the people who, as I said earlier, are at the both the beginning and the end of this journey. Yeah. And so we have done that systematically, and we've insisted that in all of our contractual and other arrangements. But I think it's important what Chris has outlined, which is to ensure that access to the research environment also has significant patient and public involvement in the decision making there.
And I think that goes sort of right upstream as well for things that we're thinking about. So Sue's mentioned a couple of times the services that exist around newborn intensive care and pediatric intensive care, and about how these individual rare diseases are quite rare, but collectively they're actually not that rare. And one of the areas that we're jointly exploring is the concept of whole genome sequencing for newborns. Huge ethical questions to be asked, addressed, discussed, huge logistical questions about how you would do that. For those who are interested in this topic, there is a session with Sarah Jane Marsh, who's chairing the NHS steering group, thinking about this later on today as part of this innovation showcase. But as part of that thinking and design piece, with participants from ScienceWise, which is a Wellcome Trust initiative, we've had a series of public dialogues with new parents, expectant parents, young people, old people, people from different regions, midwives, other groups, around this concept, both the initial concept of we could sequence someone at birth to highlight if they have a serious and early onset and treatable condition that we can then get right into treatment straight away, but also this idea of having the life course benefit of having your whole genome on your record and using that to drive wellness or early interventions in other areas and so on. Huge questions. And what I find really inspiring is that often those groups are the ones who are telling us to do it right, but to absolutely get on with it. And these dialogues are quite intense and involve a lot of those groups learning about genomics, learning about these different questions. And what we're hearing from them is, if we can do this, if we can save lives, improve lives, get on with it, just do it right and do it thoughtfully.
Yeah, absolutely. And there's a follow-on question that leads on very nicely from that, from Megan Horton, which is that some individuals and communities are less likely to access genetic testing or consent to research due to a lack of trust or communication or understanding. And how do you think we can improve this so that we can provide more equitable care to patients?
at the risk of sounding like an advert for sessions today, we have our final session of the day is on the need for more diverse representation in the genomic data sets. So this big program around diverse data. And I think it's an excellent question. And we saw this particularly acutely through COVID as well. We saw differential rates of infection, differential rates of severity in different communities, whether that was different ethnic communities, communities in highly deprived areas versus more affluent areas, and so on. The mission statement of Genomics England is bringing the benefits of genomic medicine to everyone, and that final word, everyone, is really important. If we're generating insights which are based on research which is only done on European Caucasian genomes, because those historically have been the ones who've been most of the volunteers in those kind of large-scale biobanks. There's a risk that those insights are not relevant to different groups and so on. So there's a huge amount of work to do there. We've learned a lot through COVID about the very reasonable and often specific questions or concerns that different communities have, whether that's about vaccination, whether that's about participation in research. And I think there's an increasing sort of enthusiasm and belief in the whole community that we need to kind of, to some extent, turn on its head what has historically been the approach of, well, we just need to explain this better to people. Actually, no, we need to go to where those people are, understand their concerns, which are often really legitimate, and work together to come up with good answers to that. Yeah.
So, but in the NHS Genomic Medicine Service, I mean, we undertake somewhere between 1/2 to 3/4 tests a year. That is, not at the moment in partnership with Genomics England, not at the whole genome sequencing area. That testing, by and large, is actually representative of our populations. And we have to also, it's quite complex, because all the different clinical conditions we test for, there will be a different incidence and prevalence within different populations. We know that from the 100,000 Genomes project that the participation was actually reflective of the community makeup of our population in the UK. That having been said, there's always more that we can do, and the more we need to do. But the one thing we have to do is make sure we have information that's accessible to people whose language is not English. They have to be supported by people who can actually speak and interact with them in their own language. And certainly through our NHS Genomic services, we have people who can support them, or they link with the broader support teams who support people where English is not their first language, and understand some of the sensitivities in some of the communities that make up our diverse and wonderful population in this country.
Yeah.
Thank you both very much. The next question comes from Neil Munro. I gather he's not the only person to ask this question, so definitely an important one to address. Please, can we have a little bit of information about how genomic investigation is going to address neurodevelopmental disorders, autism and the like? Are they included? So perhaps I'll start.
You start that one.
Yes, that's a really, really important question. That the conditions that you're referring to in that question are really a sort of group of lots of different conditions, and we know that some of the conditions which cause neurodevelopmental disorders and autism are conditions which are caused by a single faulty gene, and often in that situation that's a single faulty gene where the gene fault has happened for the first time in the affected child and hasn't been inherited from either parent. And that is the classic situation where the type of trio broad approach that we were talking about earlier in the conversation is the key to unlocking those. There are hundreds and hundreds of genes that can be involved in these conditions, so doing them one by one really isn't an option, and we've only really been able to offer routine diagnostic testing in this context since we've been able to do this broad trio approach. And the importance of the genome sequencing service in particular is the scalability of that service. We know that there are a large number of children with these sorts of disorders, and being able to scale that up is really important. Beyond the conditions which are caused by a single gene, there are many conditions which look very similar when we look at and talk to patients. But which are not caused by a single faulty gene, but maybe caused by a combination, a sort of bad hand of genes of much smaller effect, but going together with other influences or environmental factors which we don't fully understand. And in that context at the moment, the level of diagnostic testing that we can do is relatively limited, really because we are still understanding the genetic and genomic architecture of those disorders. So it's definitely that there are definitely a number of these conditions which are in the phase of extensive research work and which are really important to research further and to understand better. And those I would say for those disorders really the diagnostic solutions for those disorders are still in that sort of more distant pipeline that we can see coming towards us, but which aren't fully implementable in the diagnostic context until we've got a bit more basic understanding of how they function in terms of their genomic architecture. But certainly intellectual disability is an important category of a test in the National Test Directory, which NHS England have produced to describe who who's likely to benefit from genetic testing in the diagnostic context. And we've seen around a third of the children who've come through that so far in the whole genome sequencing service have a diagnosis. So those sorts of disorders are definitely a really important part of the Genomic Medicine Service and the whole genome sequencing diagnostic service within it. Would either of you like to add anything?
The only thing I was going to add, Ellen, was just around the National Test Directory. And this directory is the mandated directory for testing within the NHS. So what defines eligibility for testing, right up from single genes right through to whole genome sequencing. And the reason I was going to mention it is the way we've structured this is to be able to take evidence throughout the year and to make updates to that test directory at least annually, but more frequently if there are significant there's a significant evidence base that suggests we need to introduce it earlier. So these are the conditions that we're constantly looking at to understand whether we're close to being able to introduce whatever type of testing is appropriate.
And I think one sentence from me, and you can correct me if I'm wrong, which I might be, but I think I'm right in saying that intellectual disability is, if not the largest, one of the largest groups of participants in the National Genome Reference Library as well. So it's a very large research community out there working on a lot of these topics that we can then, through the test directory, kind of bring into clinical care.
It's also one of the areas where the research advances, I would say, are coming through most rapidly, and certainly in my own clinical practice where we've seen results coming back of the sort that we were talking about earlier, where a researcher has picked out something new or something particularly complex which wasn't picked up on the first round of analysis. It's probably the key area where those sorts of results are coming through most rapidly after the 100,000 Genomes project as well. Excellent. So perhaps we could move on now to a question from Carl. To what extent do you see a future of germline sequencing of inherited genetic profiles to predict and manage toxicity risks for individual patients or complex diseases? So I think, Sue, you talked a little bit about the DPYD program, which is within this area. But do you see that expanding and also expanding to complex diseases which aren't necessarily caused by a single genomic fault?
So if I could just stick, first of all, to sort of drug interactions. And I think what we know from what was an initial analysis of the 100,000 Genomes project participants, and around 8,000 of those whole genome sequences, is there were potentially 80% actionability in the drug gene interactions that have been looked for that might affect the way in which an individual receives that drug. It's not all about toxicity per se that leads to a severe side effect. It might be how a drug is metabolized and whether more is needed to be given, but it also gives us that adverse drug reaction, and remembering that we have about 1 in 15 hospital admissions every year associated with that adverse drug reactions. So at the moment, we still have more work to be done about how this would be applied within the NHS. We very much looked at the type of drug gene pair we might need to look at and introduce in the NHS, and we've got down to evidence that supports around 41 drug gene pairs. So it will be around classes of drugs like those to treat neuropsychiatric disease. And in NHS England, we're going to be working on a strategy for introducing sort of whole genome, for pharmacogenomic analysis into the NHS. But it will require further investment to do this over and above DPYD. And as I've said, we can start to look at how we use germline, whether it's germline sequencing, much more broadly. But as Chris has already alluded to, I think through the newborn sequencing programme, there's a real opportunity there to think about how it can be used and used in a systematic way. That having been said, we will look at this within the germline sequences that we've got within the current WGS Go Live system, and look at the evidence that we've got for the introduction. But it does all come down to evidence, and it also comes down to understanding how we're going to implement it in the NHS systematically, and not just for a few, and how much it's going to cost.
Thank you very much, Sue. I think that's an excellent way to close. That really sort of summarises our session very nicely this morning. So I'd like to thank Chris and Sue very much for all of your conversation this morning, and to thank our participants online very much for joining us and for submitting such excellent and interesting questions. Thank you very much.
Well, thank you, Ellen.
Thank you, Ellen.
Thanks, everyone.